Genetics. The study of heredity and variation is called genetics. Mendel's experiment. Mendel was very careful about the selection of the plant for his hybridization experiments. He selected the garden pea Pisum sativum as experimental plant because of the following reasons. The plant was easy to grow in pots or in open ground. It had short life cycles. The plant has self-pollinating flowers. Cross-pollination was also possible. The plant possesses distinct contrasting heritable characters. Mendel chose 7 pairs of characters for a study which is summarized in the following table. Character, dominant trait, recessive trait, character, dominant trait, recessive trait. Seed shape, spherical, wrinkled. Seed color, yellow, green. Seed color, flower color, purple, white. Pod shape, inflated, constructed. Pod color, green, yellow. Flower position, axial, terminal. Stem height, tall, dwarf. Inheritance of single trait. One of the reasons of Mendel's success was that he confined to study of limited traits in phi. In his hybridization crosses experiments, first he studied the inheritance of single trait and then added in his study more traits one by one. A cross between two individuals that differ with one particular trait is called monohybrid cross. For example, Mendel crossed a true red round seed shaped plant with true red wrinkled seed shaped plant. Such plants are referred as first parental generation, P1. In first filial generation, F1, he observed all the offspring with round seed shape. He supposed a round seed shape as dominant trait. Next, he allowed the hybrid of F1 generation, first generation, to be self-fertilized. In second generation, F2 generation, he noticed that both the parental characters appeared. The character that appeared in F1 was called the while hidden character that made its appearance in second generation was known as the recessive character. The dominant character in this case, round expressed as approximately 75% offspring, whereas the recessive wrinkled in this case appeared as only 25% of the offsprings. He also noticed that dominant character remained the same even when he conducted reciprocal cross, back cross. He tested each of the 7 pairs of contrasting characters given in the table. For the phenomenon of dominance and recessiveness, Mendel observed that the recessive character appeared in second offsprings in an average ratio of 3:1. Mendel's predictions. The science of cytology was in this primitive state during Mendel's time. However, Mendel visualized the cause of inheritance as factors or elements which were later named as genes by Johnson in 1909. According to Mendel, each male and female parent contain a pair of factors, and each parent passed only one factor of a pair of their offspring. He also predicted that each factor retained its individuality from generation to generation. The factors contributed united randomly to produce the characters of the hybrid. Thus, he indirectly predicted the reduction in chromosome number during gametogenesis and a physical hereditary mechanism. Mendel's principles of inheritance. Mendel did not publish any law as erroneously described in the various books. The rediscoveries of Mendel's work were responsible for our understanding of Mendel's principles. They left Mendel's work could be represented by laws of heredity. These laws are the laws of dominance, laws of segregation, and laws of independent assortment. Symbols used to represent mental laws. According to the classical method of symbolism, the dominant allele is represented by the capital letter while the recessive allele by the small letter. Thus, tallness will be represented as capital T, recessive character by small t. In the modified method, according to abnormal recessive allele, the symbol is chosen. For example, the condition of albinism is characterized by the lack of melanin pigment in the skin, hair, eyes, etc. This condition is a rare condition caused by recessive allele in homozygous condition. The symbol in this case is a for recessive allele and A for the normal allele. Another method is followed in wild plants, bacteria, and viruses. When out of two phenotypes, one is more common in the population than its alternative form, it is referred to as the wild phenotype. The rare form is called the mutant phenotype. The symbol plus is used to indicate the normal allele for wild type, and the basic letter is borrowed from the name of the mutant type. Love dominance. According to the law of dominance, different characters are controlled by units called factors. Factor occurs in pairs of a pair. One factor dominates the other. Law of segregation. According to the law of segregation or law of favority of gametes, in heterozygous, the dominant and recessive allele remain together without mixing with each other. The alleles separate or segregate from each other during gametogenesis so that each gamete receives only one allele, either dominant or recessive. During his experiments, Mendel encountered some traits did not follow the law he had encountered. These traits did not appear independently, but always together. But at least one other trait Mendel could not explain what happened and chose not to mention it in his work. Today, it is understood that these traits are because of alleles located on the same chromosome. Inheritance of two traits. A dihybrid cross describes a mating experiment between two organisms that are regularly different for two traits. The offspring of such cross is called dihybrid, which is heterozygous, is at two different genetic loci. After understanding the inheritance of single trait, next Mendel decided to study inheritance of two traits simultaneously. For this purpose, he performed a hybrid cross on pea plants and discovered a fundamental law of genetics called the law of independent assortment. Mendel began his experiment by first crossing two homozygous parental organisms that differed with respect to two traits. An organism that is homozygous for a specific trait carries two identical alleles at a particular genetic locus. Mendel chose to cross a pea plant that was homozygous and dominant for round, capital R, yellow, capital Y, seeds with a pea plant that was homozygous and recessive for wrinkled, small r, green, small y, seeds, represented as capital R, capital Y, cross, small y. The offspring of the capital Y cross to small y cross, which is called the F1 generation, first filial generation, were all heterozygous plants in round with round yellow seeds. And the genotype capital Y, small y. Next, Mendel crossed two plants from the F1 generation, first filial generation. This strip is represented as capital R, small r. capital Y, small y, cross, capital R, small r, capital Y, small y. Seeds, fertilization, capital R, round, yellow, wrinkled, green, round yellow, wrinkled green. This is dominant character and this is recessive character. After fertilization, second generation, seeds, capital R, small r, capital Y, small y. But seed is round. This is dominant character. Gametes, R, Y, capital Y, small y, capital R, small y, small r, capital Y. Self fertilization. F1 generation. This is crossing between these. R, R, Y, Y. Round, yellow. Capital R, capital Y, small y. Round, yellow. Capital Y, small y. Round, yellow. Capital Y. Round, yellow. Round, yellow. Wrinkled green, recessive character. Round, green. Wrinkled yellow, recessive character. Wrinkled yellow. Round yellow, dominant character. Wrinkled, this is round green because there is one dominant character, capital R. Round yellow, dominant character. Round yellow, dominant character. Round yellow. Wrinkled yellow, recessive character because there are two small r's. Round yellow, dominant character. Wrinkled yellow, recessive character. R R. You know R R Y Y. R R Y Y. Phenotypic ratio 9 round, 3 yellow, round green, 3 wrinkled yellow, wrinkled green. Mendel observed that the two progeny of his dihybrid cross 9 ratio 3 ratio 3 ratio 1 and produced 9 plants with round yellow seeds, 3 plants with round green seed, 3 plants with wrinkled yellow seeds, and 1 plant with wrinkled green seeds. Conclusion. From this experiment, Mendel concluded that the pair of traits in the parental generation assorted independently from one another. From one generation to the next, lap independent assortment. The lap independent assortment states that two pairs of contrasting traits with when followed and across, the alleles of one pair assort independently with allele of other pair. Independent assortment of genes and their corresponding traits was first observed by Gregor Mendel in 1865 during his studies of genetics in pea plants. Mendel was performing the hybrid crosses, which are crosses between organisms that differ with regard to two traits. He discovered that the combination of traits in offspring of his crosses did not always match the combination of traits in the parental organism. From his data, he formulated the principle of independent assortment. We now know that this independent assortment of genes occur during meiosis. In eukaryotes, meiosis is a type of cell division that reduces the number of chromosomes in a parent cell by half to produce four reproductive cells called gametes. In humans, diploid cells contain 46 chromosomes, with 23 chromosomes inherited from the mother and a second similar set of 23 chromosomes inherited from the father. Pairs of similar chromosomes are called homologous chromosomes. During meiosis, the pairs of homologous chromosomes are divided in half to form haploid cells. And this separation or assortment of homologous chromosomes is random. This means that all of the maternal chromosomes will not be separated into one cell, while all paternal chromosomes are separated into another instead. After meiosis occurs, each haploid cell contains a mixture of genes from the organism mother and father. Another feature of independent assortment is recombination. Recombination occurs during meiosis and is a process that breaks and recombines pieces of DNA to produce new combination of genes. Recombination scrambles pieces of maternal and paternal genes, which ensures that genes assort independently from one another. It is important to note that there is an exception to the lab-independent assortment for genes that are located very close to one another on the same chromosome because of genetic linkage, statistics, and probability relevant to genetics. Two basic rules of probability are helpful in solving genetics problems: the rule of multiplication and the rule of addition. Rule of multiplication, product rule. Rule of multiplication is that the probability that independent events will occur simultaneously is a product of their individual probabilities. For example, question: in a Mendelian cross between three plants that are heterozygous for flower color capital P small p, what is the probability that the offspring will be homozygous recessive? Answer: probability that an egg from the F1 capital P small p will receive a phi allele 1 over 2. Probability that a sperm from the F1 will receive a phi allele 1 over 2. The overall probability that two recessive alleles will unite, one from the egg and one from the sperm simultaneously at fertilization is 1 over 2 cross 1 over 2 is equal to 1 over 4. Rule of addition. Some rule. Rule of addition is that the probability of an event that can occur in two or more independent ways in the sum of separate probabilities of the different ways. For example, question: in Mendelian cross between pea plants that are heterozygous for flower color, capital P small p, what is the probability of the offspring being heterozygote? Answer: there are two ways in which heterozygote may be produced. The dominant allele Phi may be in the egg and the recessive allele P in the sperm, or the dominant allele may be in the sperm and the recessive in the egg. Consequently, the probability that the offspring will be heterozygous is the sum of the probabilities of those two possible ways. Probability is that the dominant allele will be in the egg with the recessive in the sperm is 1 over 2 cross 1 over 2 is equal to 1 over 4. Probability that the dominant allele will be in the sperm and the recessive in the egg is 1 over 2 multiplied by 1 over 2 is equal to 1 over 4. Therefore, the probability that heterozygous offspring will be produced in 1 over 4 plus 1 over 4 is equal to 1 over 2. Statistical nature of inheritance. If a seed is planted from the F2 generation of monohybrid cross, we cannot predict with certainty that the plant will grow to produce white flower. We can say that there is 1 over 4 chance that the plant will have white flowers stated in statistical terms among a large sample of F2 plants. Approximately 25% will have white flowers. The larger the sample size, the more likely it will be that the percentage of the result will approximate the prediction. Statistics and probability. Probability as the chance to an event will occur. Probability equals the case of divided by the total number of cases. P is equal to I over N, assuming all cases are equally likely events. P for heads, heads plus tail, 1 over 2. P for tail, tail over head and tail, 1 over 2. P for head or tail, 1 over 2 plus 1 over 2 is equal to 1. For head and tail, these are mutually exclusive, but head and then tail is possible. 1 over 2 multiplied by 1 over 2 is equal to 1 over 4. The sum rule, if either or then add the probabilities die. What is the probability that the die will fall on either one or two spots is equal to 1 over 6 plus 1 over 6. Product rule, the end rule multiply the probabilities. What is the probability that die will fall on one and next row? 1 over 6 multiplied by 1 over 6. Exceptions to Mendelian inheritance. Since Mendel time, our knowledge of the mechanism of genetics inheritance has grown immensely. For instance, it is now understood that in how many different ways alleles interact with their contrasting partner alleles at the same locus. These relationships between the contrasting alleles at the same locus in heterozygous state are called dominance relations. Although Mendel had observed only one form of dominance relation, complete dominance, but later on many geneticists became able to explain several exceptions to the Mendelian inheritance that could not be explained on the basis of complete dominance. These exceptions are said to have non-Mendelian inheritance patterns, where all dominance relations are of following types: complete dominance, incomplete dominance, codominance, overdominance. Complete dominance, when one allele completely dominates the other in heterozygous state, is called complete dominance. In this case, the heterozygous have the same phenotype as the homozygous have. The contrasting pair of alleles for all seven characters chosen by Mendel showed complete dominance. Incomplete dominance, when neither of the two alleles expresses independently in heterozygous state, rather a blend of expression of both alleles is appeared, is called incomplete dominance. In 1899, Carl Kern was working on a flowering plant named Poroclac mirabilis jalapa. When he crossed a true breeding red flowered plant with a true breeding white flowered Poroclac, all the F1 hybrid had pink flowers. This novel phenotype had a shade intermediate between those of the parent due to an intermediate amount of pigment in petals. When Coron self-fertilized, F1 pink and F2 showed all three phenotypes of flower in the ratio of 1:2. One red, two pink, one white. Red was homozygous for red alleles and white was homozygous for white alleles. But when allele red and allele for white were present together in the same plant, neither of them masked the effect of other. Rather, these alleles showed incomplete dominance in the form of pink flower. As there is no truly dominant allele, the usual capital and small letter distinction for dominant and recessive trait is not necessary. Both the alleles are represented by the same letter, but are numbered differently to distinguish white from red. Alleles for red as designated as R1 and allele for white as R2. Rounded square indicates that the phenotypic ratio is the same as the genotypic ratio. There is absolutely no need of test class codominance. It is not an intermediate quantitative expression like incomplete dominance, but in this case, different alleles of a gene that are both expressed in a heterozygote condition are called codominance. For example, in case of blood groups AB for antigen A and antigen B are expressed and group is named as blood group AB. Overdominance, when the phenotypic expression of heterozygotes become more intense than the homozygous state of the dominant allele is called overdominance. For example, in fruit fly Drosophila melanogaster, A heterozygote has more quantity of fluorescent pigments in eyes than wild or white-eye homozygote. ABO blood group system. Multiple alleles. Allele is the form of gene which codes for one possible outcome for phenotype. For example, in Mendel-Fee investigations, he found that there was a gene that determined the color of the pea pod. One form of it, one allele, creates yellow pods and the other from allele create green pods. It indicates that possible phenotype of one trait pod color are determined by two alleles. Form of the one color gene of a trait exists in three different phenotypes. There must be some sort of inherent interaction like incomplete dominance or codominance is found between the two alleles. Now, if there are four or more possible phenotypes for a particular trait, then more than two alleles for that trait must exist in the population. All of such alleles are called multiple alleles. Multiple alleles are produced by gene mutation. Some genes may have as many as 300 alleles, but individuals have only two of those alleles. Why? Because individuals have only two biological parents. We inherit half of our gene alleles from mother and the other half from father. So we end up with two alleles for every trait in our phenotype. An excellent example of multiple allele inheritance is human ABO blood group system. It is the most well-known blood group system, which was discovered in 1901 at the University of Vienna by Karl Landsteiner in the process of trying to learn why blood transfusion sometimes cause death and at other times save a patient. In 1930, he received the Nobel Prize for his discovery of blood types. ABO blood groups. ABO blood groups are found in all humans and in many other primates such as apes, chimpanzees, baboons, and gorillas. There are four principal types AB, and O. There are two antigens and two antibodies that are mostly responsible for ABO types. The specific combination of these four components determines an individual type in most cases. ABO blood groups. ABO blood groups. ABO blood type A antigen A antibody B donor A O B antigen B antibody A donor B O AB antigen both A and B antibodies none donors AB O O group antigen none both antibodies A and B only O. For example, the people with type A blood will have the antigen on the surface of their red blood cell as shown in the table below. As a result, antibodies will not be produced by them because they would cause the destruction of their own blood. However, if B type blood is injected into their system, anti-B antibodies as their plasma will recognize it is alien and boast or agglutinate introduced red cells in order to cleanse the blood of alien protein individuals with type O blood do not produce AB antigens. Therefore, their blood normally will not be rejected when it is given to others with different ABO types. As a result, type O people are universal donors for transfusions, but they can receive only type O blood themselves. Those who have type AB blood do not make any antibodies. Their blood does not discriminate against any other ABO type. Consequently, they are universal recipient for transfusion, but their blood will be agglutinated when given to people with every other type because they produce both kinds of antigens. O blood groups people are universal donors for transfusions. Genetic basis of ABO blood groups. Mustin explained the genetic basis of ABO system in 1925. It has multiple allelic thread that is encoded by single polymorphic gene. On chromosome number 9, this gene exists in three multiple alleles that have been produced through mutation. Gene mutation 1, mutation 2, mutation 3. Antigen A producing allele, antigen B producing allele, non-antigen producing allele. Their dominance relations are very interesting. Allele and are completely dominant over the allele I, while A and IB are co-dominant to each other because each expresses an equally in heterozygous IA-IB state to produce AB phenotype. Therefore, IA, IA or IA small I plus genotype will produce phenotype A. Similarly, IB, IB or IBI produces phenotype B. The homozygous small I will produce phenotype O. The blood group's alleles start their expression at early embryonic stage and keep on expressing themselves till death. Therefore, the blood group phenotype of person never changes throughout life. Occurrence of some other blood group systems. There are numbers of different blood group systems found in human. The International Society of Blood Transfusion has recognized up to 30 major group systems. These systems are characterized by the presence of or absence of specific molecules called antigens that are situated on the surface of the red blood cells. Most antigens are protein molecules. Two main blood group systems are ABO system and RH rhesus system, got from macaca rhesus monkey. These two systems are more significant because incompatibility between donor and recipient's blood with respect to these two systems may become dangerous to life. In the ABO system, all blood belongs to one of four major groups AB, ABRO, and RH system. There are two groups RH positive or RH negative, but there are more than 200 minor blood groups belonging to the rest blood group system other than ABO and RH system that usually do not complicate the blood transfusion. These are known as rare blood types. Example of blood group system other than ABO and RH are MNS, Philodron, Kill, Lewis, Duffy, Kidd, Digo. RH blood group system. The RH rhesus blood group system, including the RH factor, is one of the currently 30 human blood group system. It is clinically the most important blood group system after ABO. The name of this system RH is derived from rhesus monkey, Macaca rhesus, because its antigen was first discovered and added by Lensteiner in 1930 century. The Rh blood group system currently consists of 50 defined blood group antigens, among which the five antigens D, C, capital D, capital C, small c, capital E, and E are the most important ones. The commonly used term Rh factor, Rh positive and Rh negative refers to D antigen only. So the persons having this antigen are called Rh positive and those in which it is absent are called Rh negative. Other antigen of Rh blood group system have no significant role in blood transfusion. On the other hand, the D antigen incompatibility between donor and recipient can cause problem not only during blood transfusion, but it is also a relevant cause of the hemolytic disease of the newborn or erythroblastosis fetalis. ABO blood type antigen are not only found on the surface of red blood cell, they are also normally secreted by some people in their body fluid, including saliva, tears, and urine. Such persons are called secretors. Whether someone is able to secrete them is genetically controlled by dominant secretor gene SE present on chromosome number 19. Genetic basis of Rh blood group system. Rh blood group system is encoded by three genes C, D, and E, which occupy two tightly linked loci or loci. Alleles of gene D occupy one locus called locus D, while gene C and E alternatively occupy the other locus. The D locus is of prime importance because it is associated with the formation of D antigen, commonly known as Rh factor. GD has two alleles, D capital D and small d. D is completely dominant over small d. Person having genotype capital D or capital D small d have D antigen Rh factor on their RBC and are Rh positive. Person with genotype small d do not have Rh factor and are Rh negative. Maternal fetal Rh incompatibility. The Rh factor is notorious in cases where antibodies produced by a pregnant woman react with the red blood cells of her developing fetus. The situation arises if the mother is Rh negative, but the fetus is Rh positive, having inherited the factor from the father. The mother develops antibodies against the Rh factor when small amounts of fetal blood cross the placenta and come in contact with her lymphocytes, usually late in pregnancy or during delivery of the baby. Typically, the mother's response to this first exposure is mild and without medical consequences for the baby. The real danger occurs in subsequent pregnancy when the mother's immune response against the Rh factor has already been exposed, and her antibodies can cross the placenta and destroy the red blood cells of an Rh positive fetus. This condition is referred as erythroblastosis petolis. To prevent this, the mother may be injected with anti-Rh antibodies after delivering her first Rh positive baby. An erythroblast is a type of red blood cell which still retains cell nucleus. It is the immediate precursor of normal erythrocyte. The antibodies destroy the Rh positive red blood cell that have entered the mother's circulation before her own immune system has been stimulated by the Rh antigen. Gene interactions. During the study of Mendelian experiments, you have learned about the phenomenon of dominance, which is an interaction between the contrasting alleles of same locus. Now you are going to understand the interaction between the alleles of different gene pairs located on different loci or loci of same or different chromosomes. These are known as non-allelic interaction or intergenic interactions. Epistasis. An example of non-allelic interaction is epistasis, which can be defined as the phenomenon in which the effect caused by the genes at one locus interferes with or hides the effect caused by another genes at another locus. In such interactions, the gene which suppresses or masks the effect of action of a gene at another locus is known as epistatic gene, and the gene which is suppressed is known as hypostatic gene. Epistasis must not be confused with dominance. Dominance is the relationship between alleles of the same gene occupying the same locus, but epistasis is the interaction between different genes occupying different loci or loci. Following table further illustrate the difference between epistasis and dominance. Bombay phenotype. The Bombay phenotype in human is an example of epistasis. This is an altered and very rare blood group phenotype in which individuals are phenotypically O, but genotypically they may be like A, B, or AB. Actually, the production of A or B antigen also is dependent upon its substance, which is synthesized by another gene at on chromosome number 19. Its recessive allele small it does not synthesize this substance. This blood phenotype was first discovered in Bombay, now known as Mumbai, in India, by R.D. Dr. Y.M. Bendy as published in 1952. It is present in about 0.0004%, about 4 per million of the human population generally, though in some places such as Mumbai, formerly Bombay, local population can have occurrences in as much as 0.01%, 1 in 10,000 of inhabitants. The H substance is precursor to the A and B antigen. For instance, the B allele must be present to produce the B enzyme that modifies the H substance to become the B antigen. It is same for A allele. However, if only recessive alleles for H substance are inherited, as in the case of Bombay phenotype, the H substance will not be produced. Subsequently, the A and B antigen also will not be produced. The result is an O phenotype by default, since a lack of A and B antigen is the O type. This means that ABO genotype also require HH or capital H small h genotypes for their correct expression. So the individuals who are homozygous recessive at the H locus, small h will be blood type O regardless of the genotype at the I gene where the alleles for ABO blood types resides. Polygenic inheritance. Genotypes interact with environment to produce phenotypes. Phenotypic expression of the traits are up to different types, qualitative and quantitative. Qualitative traits have few phenotypes that have sharp and more obvious differences among them, so they show discontinuous variations. On the other hand, quantitative traits comparatively have large number of phenotypes that have small and less striking difference, so they have continuous variations. For instance, some traits like pea seed shape exist in two sharply distinct phenotypes, round and wrinkled. Other like 4 o'clock mirabilis jalafa flower color can have three phenotypes, red, pink, and white. Still other like ABO blood group system have four qualitative different phenotypes A, B, AB, and O. But there are number of traits such as height, weight, intelligence, and skin color in humans, and grain color and weight exhibit continuous quantitative variation over the wide range of many phenotypes from the extreme to another. Mendel focused on traits that showed only two qualitative different phenotypes, which could be determined by just two alternate alleles of a single gene. But a wide range of phenotypes of quantitative trait cannot be encoded by a single gene with two alleles. Even few multiple alleles of a single gene cannot make a large number of phenotypes. In fact, a continuously varying trait is encoded by alleles of two or more different gene pairs found at different loci, all influencing the same trait in an additive way. These quantitative traits are therefore called polygenic traits. All the genes that control quantitative trait are called polygenes, which have a small positive or negative effect on the character. Polygenes supplement each other and sum of positive and negative effect of all individual polygenes produce quantitative phenotype of continuous varying traits. Wheat grain color. Wheat grain color is a good example of polygenic multiple gene inheritance. Wheat grains show a continuous variation in color from white to dark red. Approximately 7 different types are found in wheat population all over the world. Some grains are white, some are deep red, but most grains have shades in between from light pink to moderately dark red. Nelson studied the genetics of wheat grain color, and he crushed a true breeding dark red grain plant with true breeding white grain plant. All F1 grains had light red color intermediate between two parental shades. It seemed as if it was case of incomplete dominance, but when F1 grains were grown to mature plants and crossed with each other, F2 grains had exactly 7 shades of color in the following ratio. Dark red 1, moderately dark red 6, red 5, 15, light red 20, pink 15, light pink 6, white 1. Three different genes pairs capital A small a capital B small b capital C small c at three different loci contribute to the weight grain color. Each individual would contain six alleles for the trait. Alleles A, B, and C codes for an equal amount dose of red pigment, which is positive effect, but none of A, B, and C encode red pigment, which is negative effect. If all the six allele codes for red pigment, capital A, capital A, capital B, capital B, capital C, the grain is dark red. When none of the six allele encodes red pigment, small a, small a, small b, small c, the gram is white. When a grain has one allele for red pigment, capital A, capital B, capital C capital A, small b, small c, or small a, capital B, small c, or small b, capital C, small c. Its color is light pink. If it has two alleles for the pigment, capital A, capital B, small c, or small a, capital B, capital C, small c, or capital A, small b, capital C, small c, it is pink. If it is three pigment alleles, it will be light red. Similarly, four alleles color dose will make red, and five alleles color dose will produce moderately dark red again. Thus, color phenotype of grains depend upon the number of pigments producing alleles A, B, and C. Environmental factors like light, water, and nutrients also influence the amount of grain color. Human skin color. Human skin color is a good example of polygenic multiple gene inheritance. Skin color is largely determined by the amount of melanin the skin produces. Dark skinned individuals produce more melanin than light skinned individuals. At least three genes regulate the amount of melanin produced. Gene A is involved in the permanent survival, proliferation, and migration of melanocytes. Gene B encodes the enzymes tyrosinase, which is involved in the production of melanin from tyrosine. Gene C is primarily responsible for determining whether phenylalanine and eumelanin is produced in humans. Each gene has two forms: dark skin allele represented by capital case letter A, B, and C, and light skin allele represented by small case A, B, and C. No allele is completely dominant to the other, and heterozygous exhibit an intermediate phenotype incomplete dominance. A, B, and C act as dark skin alleles and genotype, but they aid pigment by increasing melanin production. On the other hand, A, B, and C act as light skin alleles in the genotype because they inhibit melanin production. There are seven different shades of skin color, ranging from very light to very dark. Most individuals have the intermediate skin color. This later genotype would be characteristics of mulatto, offering a black and white parent. In cross between two mulatto genotypes, each parent produces eight different types of gametes, and these gametes combine with each other in 64 different ways, resulting in a total of seven skin colors. The skin color can be represented by the number of capital letters ranging from 0, no dark skin allele, to 6, all dark skin allele. The approximate shades of skin color corresponding to each genotype are shown in table 22.4. Gene linkage and crossing over. Gene linkage. The number of genes in a cell is far greater than the number of chromosomes. In fact, each chromosome has hundreds and thousands of genes located on the same chromosome that tend to be inherited together. And genetic crosses are said to be linked genes, and the phenomenon of staying together of more than one gene on the same chromosome is called gene linkage. If genes are linked on autosome, their linkage is called autosomal linkage. Similarly, if they are linked on sex chromosomes, their linkage is called sex linkage. All the linked genes found on the same homologous pair of chromosomes form a group known as linkage group. So the number of linkage groups in an organism are equal to the number of homologous pair of chromosomes in that organism. For instance, in humans, the genes for sickle cell anemia, leukemia, and albinism are found on chromosome number 11. So these traits are supposed to be in the same linkage group. As it is mentioned earlier, that linked genes tend to be inherited together, block inheritance in the offspring. So usually they do not show recombination, do not assort independently in the offspring. So the ideal Mendelian ratio of independent assortment is deviated. Detection of gene linkage. Gene linkage can easily be detected by performing a test cross between two gene pairs, the hybrid test cross, and such cross of test cross heterozygous individual for two traits F1 is bay crossed with recessive parent P1. If all four phenotypes combinations, parental and recombinants, are produced in equal 1 ratio 1 ratio 1 ratio 1 ratio, then there would be no linkage between the genes. If this ratio is deviated, more parental types and less recombinant types. This indicates the incomplete or partial linkage. But if only parental types are produced, complete or tight linkage is believed. To see how linkage between genes affects the inheritance of two different characters, let's take an example from T.H. Morgan experiment on Drosophila. In Drosophila, the shape of wing exists in two forms: normal wing shape and dominant or vestigial wing. Similarly, body color also exists in two forms. The gray body color is dominant or black body color. When Morgan made a cross between the individual having gray body and normal wings, with another individual having black body and vestigial wings, all the F1 progeny inherited gray body and normal wing phenotype. When these flies were test crossed with the F1 recessive, following results were observed. Gray body and normal wings, parental type 965. Black body and vestigial wings, parental type 944. Gray body and vestigial wings, recombinant type 206. Black body and normal wings, recombinant type 185. Since most of things had parental phenotypes, incomplete partial linkage, Morgan concluded that the genes for body color and wing size are located on the same chromosome. However, the production of relatively small number of offspring with non-parental phenotypes indicated that some mechanism occasionally breaks the linkage between the genes on same chromosome. Later on, it was discovered that crossing over at the time of meiosis gametes formation in animals and spore formation in plants is the mechanism that occasionally breaks the linkage between the genes on same chromosome. For instance, in humans, the genes for sickle cell anemia, leukemia, and albinism are found on chromosome number 11. So these traits are supposed to be in same linkage group. As it is mentioned earlier, that linked genes tend to be inherited together, like inheritance in the offspring. So usually they do not show recombination and do not assort independently in the offspring. So the ideal Mendelian ratio of independent assortment is derivative. Detection of the gene linkage. Gene linkage can easily be detected by performing a test cross between two gene pairs, the hybrid cross. In such type of test cross, heterozygous individuals for two traits, F1 is back crossed with its recessive parent P1. If all four phenotypic combinations, parental and recombinants, are produced in equal 1 ratio 1 ratio 1 ratio, then there would be no linkage between the genes. If this ratio is deviated, more parental types and less recombinant types This indicates the incomplete or partial linkage. But if only parental type are produced complete and child linkage is believed. To see how linkage between genes affect the inheritance of two different characters, let's take an example from T.H. Morgan experiment on Drosophila. In Drosophila, the shape of wing exists in two palms. Normal wing shape dominant, or vestigial, crossing over. Subsequent experiments demonstrated that the process which is responsible for the recombination of linked genes is crossing over. And crossing over, an exchange of maternal and paternal chromatid parts occur while homologous chromosomes are fed during propages of meiosis 1. The recombinant chromatids resulting from crossing over may being allied together in new combinations. So when they are distributed in different gametes, a wide variety of gametes are produced. Let's recall Morgan's experiment in which a female fly having gray body and normal wings was crossed by a male fly having black body and vestigial wings. Although most of the eggs had chromosomes when either the V positive or B parental genotype for body color and wing size, but some eggs had recombinant chromosome with B vestigial genotypes. Fertilization of these various classes of the eggs by homozygous recessive sperms produced an offspring population in which 17% exhibited non-parental recombinant phenotypes. These recombinants were the products of crossing over. Recombination frequency and genetic map chromosomes. Discovery of linked genes and recombination due to crossing over led one Morgan student, Alfred H. Stratford, to a method of constructing a genetic map, a linkage map, an ordered list of genetic loci or loci along a particular chromosome. Stratford hypothesized that recombination frequencies, which are the result of crossing over, depend on the distance between the linked genes on chromosomes. So, he assumed that far two genes are higher, the probability that crossover will occur between them, and therefore the higher recombination frequency. The recombination frequency is determined as follows: combination frequency, sum of recombinants, sum of all combination parental plus recombinants multiplied by 100. Using recombination data from various fruit fly crosses, Churchward assigned a relative position of several genes along the length of chromosome. For instance, figure 22.8 shows the Churchward linkage map of three genes, the body color, wing size, and third gene called Never. Never is one of many Drosophila genes affecting eye color. Never eye, a mutant phenotype are brighter, and the wild type color. The recombination frequency data is given below. With the help of these data, the only assumption about location of these three genes can be made is that CN is located midway through the B and Stratford express the distances between genes in map units. Sex determination. Whether we are male or female is one of our more obvious phenotypic characters, although the anatomical and physical differences between women and men are numerous. Chromosomal basis for determining sex is rather simple. The search for mechanism of inheritance of sex started after discovery of Mendel work. In 1900, a clear picture of the genetic basis of sex determination emerged after the discovery of sex chromosomes. Pattern of sex determination. There is a wide variety of sex determining mechanism, but three patterns are more common. X naught, XX type. This pattern of sex determination is found in grasshopper and protector bug. Male is X naught because It has only one X, X0, or mean X0. The outer sex chromosome is the other sex chromosome is missing entirely. Male is heterogametic because it forms two types of sperm. Half the sperm have X chromosome and the other half is without sex chromosome. A gamete without any sex chromosome is called null-gamete. Female is XX because it has two X chromosome. It is homogenetic. As X forms only one type of egg, every egg carries X chromosome. Sex of the offspring depend on the kind of sperm that fertilizes the egg. If an X carrying sperm fertilizes the egg, an double X female offspring is produced. If the null sperm fertilizes the egg, an X naught or X zero male offspring is produced. Sex ratio between male and female offspring is 1:1. XY XX type. This pattern of sex determination is found in Drosophila, human, and many other organisms. Male is XY and female is double X. Male being heterogametic produces two types of sex determining sperms. Half the sperm carry X chromosome and the other half carry Y chromosome. Chances for both types of sperm are equal. Female being homogametic produces only one type of egg, each with an X chromosome. Sex of the offspring is determined by type of sperm. If an X-carrying sperm fertilizes the egg, the zygote will be double X and female offspring is produced. If Y-carrying sperm fertilizes the egg, the zygote will be XY and the male offspring will be produced. The sex ratio between male and female offspring is 1:1. Sex ratio indicates chances of the sex of the offspring. Chances for a son or daughter in human birth rate are equal. XXXY type or WZZ type. This type of sex determination pattern is common in birds, butterflies, moths. It was discovered by J. Seller in 1914 and moth. It is reverse of XYXX system. Here the female is heterogametic XY, but the male is homogametic XX. Female produces two kinds of eggs, X and Y, in equal proportion. All sperms are alike, each carrying an X chromosome at its kind of egg that determines the sex of offspring. When an X carrying egg is fertilized by the sperm, a male offspring is produced, but when a Y carrying egg is fertilized by the sperm, a female offspring is produced. Sex ratio is 1:1. Comparison of chromosomal determination of sex between Drosophila and humans. Although both Drosophila and humans follow the same XY sex determining pattern, yet there is basic technical difference between the two. Presence of SRY gene on Y chromosome is essential for triggering the development of maleness in humans. Absence of Y chromosome simply leads to the female development path X0 Turner syndrome in human produced through non-disjunction, a sterile female, but in Drosophila X0, a sterile male. Similarly, XXY individual produced through non-disjunction gametes in human a sterile male called Klinefelter syndrome. But the same XXY set of chromosome in Drosophila produces a fertile female. There is close genetic balance between genes of different chromosomes. Drosophila has an X chromosome, autosome balance system. Its Y chromosome appears to have little influence on sex. Here, actually, the X chromosome is female determining and autosome are male determining. Sex of an individual depends more on the number of X chromosomes relatively to the number of sets of autosomes. An X a ratio of 1.00 or higher produces female, whereas an X ratio A ratio of 0.5 or lower produces males. Sex linkage in Drosophila. Sex linkage. T.H. Morgan made the first demonstration of sex linked trait in 1910 through his experiment on Drosophila. The common fruit fly, Morgan showed that the inheritance of eye color and sex color in a codominance fashion. He reasoned correctly that the eye color gene is located on the X chromosome but is not present on the Y chromosome. This means that the recessive allele specifying white eyes is always expressed in males but could be masked by the presence of dominant red eye color allele in heterozygous females. Drosophila melanogaster eye color turned out to be most informative trait. At first, all the flies Morgan raised were wild type for eye color. They had brick red eye wild type. In 1910, a white-eyed male appeared in a laboratory bottle. Apparently, the variant form arose through spontaneous mutation on a gene controlling eye color. Morgan established true breeding traits of white-eyed males and females. Then he did a series of reciprocal crosses represented in the following figures. Morgan experiments were designed to test protection derived from his hypothesis that factor for eye color associated with X chromosome, a trait whose gene is present on X chromosome is called X-linked trait, and a gene which is present on X chromosome with no counterpart on a Y chromosome is called X-linked gene. Same is the case with Y. Linked genes, Y chromosome do carry some genes having no counterpart on X chromosome. For example, S or Y genes on Y chromosome of men determine maleness. Y-linked genes directly pass through Y chromosome from father to son. Types of sex-linked traits. A trait whose gene is present on X chromosome is called X-linked traits. X-linked traits are commonly referred to as sex-linked traits, a gene present only on X chromosome, having no counterpart on Y chromosome called X-linked gene. Sex-linked gene inheritance follows a very specific pattern as a son inherits his X chromosome only from his mother, and daughter gets an X chromosome from each parent, an X-linked trait passes in a crisscross fashion from maternal grandfather P1 through his daughter F1 to the grandson F2. It never passes direct from father to son because a son inherits only Y chromosome from father. Sex linkage in humans. Humans have many X-linked traits, of which some, like hemophilia and color blindness, are recessive, while others are hypophosphatemic or vitamin D resistant rickets are dominant. X-linked dominant is trait which is determined by an X-linked dominant gene, while X-linked recessive is a trait that is determined by an X-linked recessive gene. Their pattern of inheritance are very different from each other. Experimental matching are not practically possible in humans. This is main important point. Mode of inheritance of human traits can be traced through pedigrees. Sex-linked recessive inheritance. Characteristics of X-linked recessive inheritance. Female possessing one X-linked recessive mutation are considered carriers and will generally not manifest clinical symptoms of the disorder. All males possessing an X-linked recessive mutation will be affected male have single X chromosome and therefore have only one copy of X-linked genes. All offerings of a carrier female have 50% chance of inheriting the mutation. All female children of an affected father will be carries. Daughter possess father X chromosome. No male children of an affected father will be affected son do not inherit their father X chromosome. Some example of X-linked possessive disorder. Some sex-linked recessive disorders: hemophilia A and B, color blindness, diabetes insipidus, X-linked dominant inheritance. Characteristics of X-linked dominant inheritance: a male or female child of an affected mother has 50% chance of inheriting the mutation and thus being affected with the disorder. All female children of an affected father will be affected. Daughters possess their father X chromosome. No male children of an affected father will be affected. Sons do not inherit their father X chromosome. Some example of X-linked dominant disorder: Alport syndrome, Coffin-Lowry syndrome, idiopathic hypophyrathyroidism, vitamin D resistant rickets, Y-linked inheritance. In mammals, Y-linked refers to when a phenotypic trait is determined by an allele, a gene, on the Y chromosome. It is known as palindromic inheritance. The Y chromosome is small and does not contain many genes. Therefore, few traits are Y-linked and Y-linked diseases are rare because the only humans which have a Y chromosome are males. The genes are simply passed from father to son with no interchromosomal genetic recombination. Chromosome Y detection are frequent genetic cause of male infertility. Another example in human of Y-linked trait was thought to be hairy hairs. It may be also be sex limited. However, this has been discredited. Genetics hemophilia. As well studied sex-linked trait in human is hemophilia, a disorder that renders the individual less able to form blood clots. This is a serious medical problem because in hemophiliacs or bleeders, even a minor injury can result a major loss of blood because the hemophilia allele is recessive and carried on the X chromosome. Hemophilia is predominantly a male disorder. In fact, hemophilia is extremely rare in females because there are so few hemophilic males that survive to marry and reproduce. Largely, the phenotypically normal female carrier maintain the faulty allele in human population. Genetics of color blindness. Red-green color blindness is a recessive sex-linked trait that renders individuals unable to distinguish shades of red or green and both appear gray. To understand why color blindness occurs more frequently in males, let us examine the type of parental combination that can produce color blindness in sons and daughters. A son inherits the color blind allele from his mother, who may be either colorblind herself or more likely a normal sighted carrier. There is a 50% chance that a son will inherit the colorblind trait from a carrier mother, whether or not the father is colorblind. XY has no bearing because a son receives only the Y chromosome from his father. For a daughter to be colorblind, however, not only her mother must have at least one allele for colorblindness, but her father must be colorblind. As this combination of parents occurs rather frequently, infrequently, there are few colorblind females in the human population. Sex-related traits. Sex-related traits are those which are associated with maleness or femaleness. These traits are not necessarily being sex-linked. These traits may be controlled by sex-linked or autosomal genes. Sex-limited traits. Sex-limited trait is a type of sex-related trait which is confined to only one sex due to anatomical differences. Such traits affect a structure or function of the body present in only males or in only female. For example, genes for milk yield and dairy cattle affect only cows. Similarly, beard growth in human is limited to men. A woman does not grow a beard herself, but she can pass the genes specifying heavy beard growth to her son. Sex influence trait. Sexual fluid traits are also a type of sex-related traits. These occur in both males and females, but they are more common in one sex. Such traits are controlled by an allele that is expressed as dominant in one but recessive in the other. This difference in expression is due to hormonal difference between the sexes. For example, baldness is sex-influenced trait. Many more men than women are bald. It is inherited as an autosomal dominant trait in males, but as an autosomal recessive trait in females. A heterozygous male is bald, but a heterozygous female is not. A woman can be bald only when she is homozygous recessive. Key points. The tendency of individual to resemble their parent is known as heredity. The science which deals with the mechanism of heredity and variation is called genetics. Mendel was the first who successfully explained the mechanism of inheritance during his research work on pea plant. According to the law of dominance, different characters are controlled by units called factors. Factors occur in pairs, and a pair of one factor dominates the other. And a hybrid cross describes a mating experiment between two organisms that are particularly different for two traits. The offspring of such cross is called the hybrid, which is heterozygous at two different genetic loci. Overall dominance relations are complete dominance, incomplete dominance, codominance, and overdominance. ABO blood groups are found in all humans and in many other primates such as apes, chimpanzees, baboons, and gorillas. There are four principal types A, B, and O. There are two antigens and two antibodies that are mostly responsible for the ABO types. The RH blood group system currently consists of 50 defined blood group antigens, among which the five antigens DC, CE, and E are the most important ones. Epistasis is defined as the phenomenon in which the effect caused by the gene at one locus interferes with or hurt the effect caused by another gene at another locus. A continuously varying trait is encoded by alleles of two or more different gene pairs found at different loci, all influencing the same trait in an additive way. These quantitative traits are polygenic traits. All the genes that control a quantitative trait are called polygenes, which have small positive or negative effect on the character. Human skin color is a good example of polygenic multiple gene inheritance. The phenomenon of staying together of more than one gene on the same chromosome is called gene linkage. In crossing over, the exchange of maternal and paternal chromatids part occurs while homologous chromosomes are paired during prophase of meiosis I. There are a wide variety of sex-determining mechanisms, but three patterns are more common, which are X0, XX type, XY, XX type, and XX, XY type, or WZ, ZZ type. A trait whose gene is present on X chromosome is called X-linked trait. A gene which is present on X chromosome with no counterpart Y chromosome is called X-linked gene. Same is the case with Y-linked genes.